Chapter 7 discusses inheritance of genes located on the same chromosome, which we call linked genes. So if chromosomes carry multiple genes, inheritance of these genes will not strictly follow Mendel's principle of inheritance. So in this chapter, we will first discuss and compare inheritance of linked gene with the inheritance of genes which are sought independently. Afterwards, we will discuss how can we predict inheritance of linked genes, and finally, we will talk about the method of determining the chromosomal location of genes and their relative order on the chromosome. First, we will compare and discuss the inheritance of two linked genes with the inheritance of genes that are sought independently. So let's first start with classical Mendelian inheritance and follow two genes, genes A and B. What Mendel stated, according his two basic principles, let's consider first principle of segregation. According to this principle, two alleles of one gene will segregate independently of each other. So allele 2 dominant A will segregate, 2 recessive A here will segregate. The same apply for B. Now, independent assortment in combination with segregation allows independent assortment on combination of alleles from two different loci. What does it mean? Look, let's follow this cross. You have parental generation when you have a homozygous dominant parent and homozygous recessive. So each of them can form only one type of gamete, parental gametes. Now, upon fertilization, you have heterozygous, double heterozygous of one generation. Now, here, what does it mean if we follow principle of segregation? That basically allele A dominant segregate from recessive allele, okay? Now the same apply for B, allele B, dominant allele segregate from recessive allele. Since if we consider that these two genes are located on two different chromosomes, this chromosome, you recall, can align randomly in metaphase plate and separate randomly during anaphase 1. So you can have combination of gamete containing dominant A with dominant B, but also with recessive B. The same apply with A, recessive A, and its combination with dominant or recessive B. So what does it mean? If you take independent assortment, you will see that half of the gametes will actually have original combination allele. We say they have parental non-recombinant combination of alleles. However, half of them which will form new combinations which you didn't find in parental generation. You don't find these two combination in parental, and we talk about this new gamete, new combination as a recombinant gametes. So recombination is not just crossing over. Recombination is sorting of allele into new combinations. So during independent assortment, you will see here you will have 50% or 1/2 original parental gametes, and we will have 50% or 1/2 recombinant gametes. So now what about linked gene? However, before we go to linked genes, I would like to point at one situation that, or one fact, that these are gametes of one parent. Second parent, if you cross two heterozygous, double heterozygous, so you know that afterwards in F2 generation, your ratio will be 9 to 3 to 3 to 1. So let's now talk about linked genes. We know that we have much more genes than we do have chromosomes. So obviously there are genes which have to be located on the same chromosome. And those genes which you find on the same chromosome we call linked genes. And since they are linked, they cannot assort independently. Their behavior depends on each other because they sit on the same chromosome, and we say that they co-segregate. Now you have one of the first examples which were described, example of linked genes outlined here, in which the authors crossed purple flowers, this is sweet peas, where purple flowers and long pollen is dominant over red flowers and round pollen. So color of flower and shape of pollen are two genes which are located on the same chromosome. So if you cross two purebred plants, you get F1 generation which resemble a dominant parent, which up to now phenotypically this cross resemble typical Mendelian inheritance or follows Mendelian inheritance. However, if you look at F2 generation, you notice immediately difference between independent assortment and linked genes. In independent assortments, the ratio we would obtain in F2 would be 9 to 3 to 3 to 1. But if you look here, if these two gene for color and shape of pollen are linked, you see that majority of parent resemble dominant parent. You have most of the dominant purple flowers with long pollen. Second, most numerous group or largest group are red flower plants with round pollen, which resemble recessive parent from parental P generation. Now recombinants, new combination of allele, you can see here, and they are least frequent. So you see immediately that two linked genes do not follow the same rule and ratios. You don't get the same ratio as during independent assortment, and the most frequent phenotype are the parental phenotype from P generation, and the least frequent are recombinant. And it comes with the mechanism how recombination can happen here only due to crossing over in meiosis 1. You remember? In meiosis 1 prophase, when two homologous chromosomes align and form tetrad, that's when crossing over take place, and that's a crossing over only which can create recombination. However, sometimes two genes sit so close together that we say that they are completely linked. Complete linkage is situation when two genes are so close that no crossing over, no recombination happens. So in that case, you will have only parental phenotypes. So only mechanism by which you can obtain recombinant gametes between two linked genes are when crossing over take place. So the best way how to define or see the difference between linked genes and genes which assort independently is to compare independent assortment and complete linkage. However, before we go there, we have to define notations for linked genes. Why it is important? Because if you follow crosses with linked genes, you must know not only genotype of the crossed individuals, but also the arrangement of the allele of two different genes. So how do we notate for linked genes? You remember we have two homologous chromosomes in one chromosome pair. One is from father and one is from mother. Now let's consider two genes on this homolog. On one locus we have gene A, and in another locus we have gene B. So this is a standard notation for linked genes. You can simplify it by using only single line, and it notates the same, two linked genes. Even more simple notation is if you just used slash. In this case, if you see this notation, you will know that two dominant alleles are located on one chromosome and two recessive alleles are located on second chromosome. One important thing I want you to note, each gene has own locus or is localized on the same place on the chromosome. So this is locus for gene A and this is locus for gene B. So you have to always annotate them together. Let's say if you have this two linked gene A and B and you annotate it this way, what would this mean? It would mean that the dominant allele A and recessive allele B share the same locus, which is not true. So be careful how do you notate your linked genes. Now, why arrangement of the gene on chromosome is important? Let's consider gene A and B with two different arrangements. Let's say one arrangement you have dominant allele on one chromosome and recessive on another, and on second chromosome you have actually one chromosome carry dominant A, recessive B, and second one carries recessive A and dominant B. If you look phenotypically, these two individuals have both wild type or dominant phenotype. So what kind of gametes they can form if we consider that gene A and gene B are completely linked? So if there is complete linkage in anaphase 1, this one chromosome will go to one gamete and the second chromosome will go to another gamete, because if there is complete linkage there is no crossing over. So this parent would only be able to form two kind of gametes. One gamete will have both allele dominant and another gamete will carry both recessive allele. In this case, in this arrangement also, if there is complete linkage, each chromosome, this chromosome goes to one gamete, this chromosome to another gamete. So the offspring, the gametes, one gamete would carry dominant allele A and recessive B, and second gamete would carry recessive A and dominant B. So now imagine these two parents are phenotypically identical. However, if you consider fully linked genes, complete linkage, and you perform test cross, you remember you test cross with homozygous recessive, you will see that offspring of these two parents will be different. And it's why arrangement of the gene on chromosomes when the genes are linked is very important. So we know already how to notate linked genes. So let's now consider complete linkage versus independent assortment. So how do you define whether two genes are linked or whether they assort independently? The best way to reveal that is to use test cross, where you cross, of course, tester homozygous recessive with heterozygous plant. So here is an example which outlines two possible scenarios. It crosses tomato plants where normal leaves are dominant over mottled leaves and tall plants are dominant over dwarf. So these two scenarios we will consider. What kind of outcome you will have if you consider completely linked genes and genes assorting independently. So let's first consider independent assortment. If you test cross heterozygous plant for normal leaves and tall, you know that if these two genes, gene for heights and gene for leaf shapes, will assort independently, so in such scenario they would form four different gametes. We already discussed this. Half will be non-recombinant and half will be recombinant. Since you test cross plan with homozygous recessive, so only gametes which tester can form is two recessive allele, you will obtain offsprings where you will have half non-recombinant and half recombinant. Within each group, you will have equal number of dominant recessive and equal number of each recombinant. So this is outcome which you get with test cross if two genes are unlinked. So you will get ratio 1 to 1 to 1 to 1. Now, what if you test cross plan where you consider these two genes as a link, as you consider complete linkage? You recall, if you have completely linked genes, they will assort only by co-segregation. There will be no crossing over. So in this case, only gametes which you will form are gametes which will contain only dominant or only recessive allele. So in test cross, what you will obtain in as a offsprings will be half of the plants which would be dominant, tall and with normal leaves, and half will be which will be recessive for both traits. So both of these offsprings resemble parental generation. So with complete linkage, you will only get non-recombinant progeny. So this was just an example. Complete linkage is rare in real life. So usually you obtain non-recombinant and recombinant progeny even with linked genes because of crossing over. Let's now consider what happens at the molecular level, at the chromosomal level, when two genes are linked. We will first consider situation under which two genes are completely linked, so no crossing over takes place. If you have gene A and B, like here, these two genes, consider that they are located on the same chromosome, and we have a homolog pair, one from father and one from mother. So you see that father is homozygous dominant for both genes and mother is homozygous recessive for both genes. If we consider no crossing over taking place and you go back and look at the meiosis 1 and meiosis 2, what you can see that under such scenario there are only four different gametes. There are four different gametes with only two combination of alleles which can take place, and these two combinations are parental combinations. So under this scenario, when there is no crossing over, the gamete that this individual can form are only non-recombinant gametes which contain original combinations of allele. Now, as we said before, this is very rare. Usually crossing over takes place in meiosis 1. So if we consider crossing over between two non-sister chromatids in the homologs, what would happen under this scenario is outlined right here. Look at the chromatids which didn't underwent recombinations. These two chromatids will give rise gametes which will have parental combination of allele. We said they are non-recombinant. So this will be non-recombinant and this will be non-recombinant. However, gametes form from chromatid which underwent crossing over will be recombinant, and they will form gametes. This recombination will end up in gametes which possess or carry new combination of alleles. They are recombinant. Now, did you notice if a crossing over take place between two non-sister chromatids, you will have 1/2 or 50% gametes which will carry non-recombinant alleles, and 50% of gamete which would carry no new combinations. They will be recombinant. So frequency of recombinant gametes under this scenario will always be half of the frequency of crossing over, and therefore the maximum proportion of recombinant gamete will be 50% if crossing over takes place between two non-sister chromatids. Now, there is another thing I would like to point out. So, if crossing over takes place in every single meiosis, you will obtain 50% non-recombinant and 50% recombinant gamete. If you recall, this is a ratio or percentage of recombinant gamete which you receive or obtain after independent assortment. So, in such scenario, it is impossible to define or determine position of two genes on the same chromosomes. It is impossible to determine percentage of recombination. So, is it possible to have 50% recombination? Yes, it is. It is possible if two genes are located on the chromosome far apart, which assure that during every meiosis there will be crossing over taking place. And we talk about if this happen, we refer to such group as a different linkage group, or that these two genes are in different linkage groups.